Hi, and welcome to the Canine Health Summit, Puppy DNA Testing. I'm Lisa Peterson from Embark Veterinary, and I'll be your host for today's event. This virtual summit is presented by LabRoots. To learn more about us, visit Embark for Breeders at our website, embarkvet.com/breeders. We have a great group today of breeders and dog lovers from around the world. Over 780 people have registered to join us. There's a great mix of attendees from people who are new to breeding to those who have been breeding for more than 20 years. I saw a lot of rare breeders who are joining us, breeds like the Stabbyhund, the Niederlander Quakerhundje, the Coton de Tillier, and even a Laponian Herder. Also, there's popular dog breeders joining us with poodles, labs, Goldens, German Shepherd dogs, Frenchies, Golden Doodles, and also many from service dog organizations. So welcome, everybody. Before we get started, I have a few housekeeping notes. We encourage you to participate today by submitting any questions during the presentation. You can type them into the Ask a Question box, which is located on the left-hand side of your screen, and click Send. We'll answer as many questions as possible at the end of each presentation. Any that we don't get to, we'll endeavor to answer via email after the summit. One note, please do not ask questions about specific dogs or litters that we don't have firsthand knowledge of, so keep the questions broad enough to benefit the most attendees. Also, if you have any technical questions, we have Noah today at the help desk. You can reach him either in the help desk room or by using the Ask a Question box during the session, and he will assist you directly. You can also find out who's here today with the Who's Here button on the top right-hand side of your screen in whatever room you're in. You can search for attendees via the whole event or who's in the room with you currently. You can request a chat by clicking on the chat icon bubble. To find your private messages, you can go to the upper right-hand corner circle with the down arrow icon to find your messages. You can also update your profile photo on the far right, top right of the teal bar. You can put yourself or a photo of your cute puppy in there. Feel free to interact with others during the breaks and also after the summit, because this platform will be live for the next 12 months. If you're so inclined to have a social media hashtag, ours is #PuppyDNATesting. And for complete bios of our speakers, please visit the speakers tab in the agenda in the auditorium room. So let's get started. Our first speaker is Dr. David Haworth, who will present New Technologies on the Horizon. David is the Executive Vice President at Embark Veterinary. He's responsible for research and development, business development, and corporate strategy, as well as a highly sought-after advisor and consultant to some of the largest and smallest organizations in the animal health and wellness space. He serves on numerous boards, including RxActuator, Dog TV, and Canine Biologics, and he chairs the scientific working group of the Human-Animal Bond Research Institute. David, take it away.
Lisa, thank you so much, and welcome everybody. Good morning for those in the US, for those around the world. Good afternoon and good evening to everybody. As Lisa said, my name is David Haworth. I'm fortunate enough to be responsible for our science and business development activities at Embark Veterinary. I am a veterinarian. I also hold a PhD in reproductive physiology. After spending a little time in private practice, I spent 11 years at Pfizer Animal Health, now called Zoetis. I was lucky then and after that to spend 4 years as the president and CEO of Morris Animal Foundation, and then another 4 years as president of PetSmart Charities in the US and Canada before I co-founded a precision medicine cancer diagnostic company called Vidium Animal Health, which last year was acquired by Anavive Animal Health, which allowed me to join my friends at Embark to do some special and hopefully impactful things in the coming years. Today, the fun thing that I get to do is introduce today's lineup of speakers and do my best to set the stage for what you're going to see. If this was a comedy show, I guess I just get to be the warm-up act. Before I do that, first I want to thank you. Someone once told me that in this life, we only have 3 kinds of assets: time, talent, and treasure. It's been my experience that of those, I can always train to learn new talents, and I can usually generate more treasure. But time is the one thing we never get more of. So that makes you spending your time with us today incredibly valuable. So that's just me calling out how much we appreciate you giving us that precious gift of your time. So for my time today, I'd like to do 2 separate but sort of related things. First, I'll speak a little bit about why we're all collected together today, what's behind this human animal bond. And secondly, I'd like to make sure we're all on the same page when it comes to the science at the heart of genetic testing. I wouldn't worry about it. It's a short science lesson that hopefully will pave the way for the really smart people who are going to be presenting to you later. So that's it. Human animal bond, history and basics of molecular biology, hopefully all in the next 16 or so minutes. So it may seem funny to talk about the human animal bond and science at the same time, but about a decade ago, several friends and I founded the Human Animal Bond Research Institute to put some resources behind the understanding of why we feel, how we feel about these miraculous creatures that share our homes with us. But also importantly, to take away some of the excuses of those who deny access to support animals in some of our most critical places that we need their special kind of magic, places like hospitals and nursing homes. But we have to recognize the fact that the role pets play now in our society is relatively new. It was only in the 40s and 50s that the very best a dog could hope for was a doghouse in the backyard, maybe a bird for a best friend. And if they ever did get into the house or give Lucy a lick on the face, the results were dramatic and bad for the dog. As society changed in the 70s and 80s, Scooby showed us that a dog could be part of the gang. He was usually understood when he communicated. He almost always got to sit in the middle seat in the mystery machine. And in more modern times, for the past 14 years, Brian Griffin is an integral part of the family in the show Family Guy. He walks on two legs. He drives a hybrid. He's a published author and is probably the only thing keeping the baby in the family, Stewie, from accomplishing world domination. The bottom line is that in the past 75 years, dogs and other pets have moved into our lives in ways our grandparents could not have been able to predict. Thanks to many things, including palatable and convenient food, effective and economical antiparasitics, and an explosion of pets in the media reflecting the role that dogs play in society. Dogs have gone from something that was once, not too long ago, restricted to the backyard to fully integrated family members that spend most nights on our pillows. But why is that? What are the driving forces that have caused this revolutionary change? And I realize that for some, putting words to something as beautiful as the human animal bond can feel maybe clinical or cold. And let me assure you that's not my intention. There are entire books written on the human animal bond and what draws us in particular to dogs and cats. Why have these creatures taken this dominant position in our lives and our society? Now, everybody is unique and every relationship between a dog and their family is equally unique. But there are four primary drivers that define why we want these animals in our lives. First is companionship. Humans are a social species. We don't love being alone. And dogs are in many ways the perfect companion. They're patient and loyal. They are the most empathic non-primate studied, meaning that their actions actually demonstrate that they get us. While I write here that pets are a surrogate for other humans, they are often, and if we're going to be honest, an improvement. Don't misinterpret me. I actually do like humans. I've been married to one for 35 years, and I helped her raise two other ones that are good and kind young adults, but I often prefer to spend my time with dogs. The second driver is our inherent need to nurture something. We see this across society. We need to care for and protect something else that's alive and responds to our care. Plants can serve the same purpose, but dogs and cats are particularly good at making us feel great about doing those things they actually require us to do if they're going to thrive. Few people don't love it when their dogs do the food dance or get excited about an upcoming walk. It's a magic feeling when we get to be able to take care of them. The third factor is an external one, and that is that pets show us and those around us that we have worth. When we come in the door and they greet us like we're the best thing that's ever walked on 2 or 4 feet, it triggers deep and important emotions around self-worth. Not only that, but dogs especially provide tangible evidence to the broader world that we have value. As Bill Murray said, I'm suspicious of people who don't like dogs but I always trust a dog when it doesn't like a person. In another example, approaching a stranger sitting on a park bench is usually considered awkward unless that person has a dog. Dogs lower the energy required to interact with someone else, acting as a catalyst for social interactions. The fourth driver of the human-animal bond is nature. There is something psychologically and spiritually important for humans to be connected to the natural world. That connectedness is met by having what was once a wild animal only a few thousand years ago laying in your bed, on your feet, or on your couch. These four drivers are the primary reasons we love having dogs and cats in our lives. They're not the only ones, and different people put emphasis on different of these, but most people can see some sense of truth in this list. I'm sure at least some of you are thinking by now that you stepped into the wrong summit. Is this not a summit on the genetics of puppies? Am I not supposed to be talking about technology? And I will, I promise, starting on the next slide, but I find it important to start a day of science with at least a small exploration of why we need that science, why we do this stuff in the first place, and why is it important? None of us would do this work or follow these paths for frozen waffles. Dogs are special. Animals are special. And the bond that we have with them is something that can be beautiful and powerful. Plus, frozen waffles never curl up on your feet on a cold winter's night. And if they did, I don't think it would be very comfortable. So let's transfer to the science. You'll hear a lot about the science behind genetic testing today from some really smart people. I thought it would be a good idea to begin the day by grounding us in some truths that might help frame what we're discussing. So if you'll let me, I'd like to talk for a few minutes about what we call the central dogma of molecular biology. This is the model that all of you have heard about, but it's probably worth reinforcing here at the beginning of the day. DNA is a basic building block of life as we know it. All living things on Earth have as their code keeper DNA, or deoxyribonucleic acid. That DNA, made up of four bases, adenine, thymine, guanine, and cytosine, in three base long letters that encode each amino acid, which are the building blocks of protein, that in turn relate the sequence of every protein that makes up an organism. In cells that have nuclei, meaning most plants and animals, they do that through a temporary formation of an RNA molecule, that second line, through a process called transcription that takes place inside the nucleus of the cell. Then the RNA molecule moves outside the nucleus, where it's translated into a protein. The proteins do everything else. They move things, they form the backbones of all the structures inside and outside of the cell. They make enzymes that allow chemical reactions to take place, and they even form the sensory molecules that allow us to perceive the physical world. Proteins are amazing. And then when they're no longer needed, many of them get broken down to metabolites. Why is this important? Well, because we have in the last 70 years found ways to harvest information at each one of these steps in order to find out things about the cell, the organ that the cell is in turn, the organism that's made up of those organs. And while there are still mysteries, an incredible number that we'll continue to explore, we really do know an incredible amount about the way these animals work at a fundamental molecular level. So these two fashionable and handsome young men are James Watson and Francis Crick, who in 1953 proposed the structure of DNA that they're pointing out here in this wire and ball model that stood the test of time and scientific inquiry. Before this point, it was widely held that DNA was just too simple a molecule to possibly hold all the information required to build the wide variety of species which inhabit this living world. But these two scientists proved everyone wrong, and since then we've expanded on those findings exponentially. And thanks to techniques like X-ray crystallography, we can now say without a doubt exactly what DNA looks like, how it's packaged, and even what molecules, usually proteins encoded by that DNA itself, that interact with the DNA look like. Like this illustration of a DNA polymerase sitting on top of a DNA strand. This incredible protein, the big purple blob, travels down the approximately 6-foot-long strand of DNA in a cell and faithfully reproduces it into 2 nearly identical strands. Just think about that for a second, if you would. First of all, DNA is so thin, it's about 2 nanometers wide, that literally feet of it can be packaged inside every single cell in the body. And this polymerase can go its entire length and reproduce that DNA with an error rate of only about 1 in 10,000 to 100,000 base pairs, which is incredible. But it's actually this error rate, which is what we call a Goldilocks error rate. It's not too many errors, but it's also not perfect, that allows for variation in nature and creates the basis for all the tests we're going to talk about today. And it's not only the DNA polymerase that we can visualize. 2 years ago, a researcher named Drew Berry created these animations that have almost perfect fidelity to the process of RNA creation. Remember, DNA gets transcribed to RNA, which then gets moved out of the cell to be translated into proteins. So in this animation, here are transcription factors assembling like the Avengers to literally cause the DNA to bend, and then when activated, shoots off this RNA transcriptase protein, the blue blob that's running down the strand of the red RNA, that unwraps the DNA helix, assembles an RNA strand, and then reattaches the DNA, which is going at roughly, in real life, the speed of a jet turbine. Base pairs are assembled with total accuracy from the nucleoplasm, and then the transcriptase cruises down the DNA like something in Star Wars, pulling in pieces and assembling them perfectly until eventually it will get to a stop codon and release the RNA strand, the yellow, into the nucleoplasm, where it gets shuttled out of the nucleus to be made into a protein. And here we have it floating through the nucleoplasm towards something called a nuclear pore and out into the cell cytoplasm. I love that. So the point is, we know a lot about the way these cells work, the way the cells divide, and the way they're organized. We use that knowledge to give you incredible insights into your dogs, their ancestry, their health conditions, and their physical traits. But why do we all know that now? And quite simply, I'll tell you, it's the economics of the technology. At the turn of the century in 2001, it was so expensive to sequence DNA to get this entire book of code assembled. It took the US government and several $billion to get the first one. But over the past 25 years, the cost of doing that has plummeted. It's dropped faster than Moore's law that says computing power will double every 2 years while the cost will go down. It's an exponential curve, so those lines aren't decreasing one at a time, they're decreasing by a factor of 10. And the cost of sequencing is actually going faster down than the exponential curve. With reduced costs, we have an increased ability to glean insights and an expanded ability for us to understand the ways the cells work. And that increased ability to understand and get information at economical costs have resulted in the incredible, powerful technology and insights that we'll be sharing with you today. And there's no sign of this trend stopping or reversing. In the coming years, we'll learn more about genes themselves, all these amazing variations that exist in nature, all the ways that genes get used, get adapted, and modified. More than that, we'll learn about the ways that genes interact with one another, how 2, 3, or even a dozen genes might exert control over complicated conditions, things like behavior, how these genes accentuate, mitigate, suppress, or enhance one another. And thanks to advances in computing technology, we can do that in ways human brains just don't have the capacity to understand. We can utilize vast data sets that go beyond anything a human could contain at once and perceive patterns more complex than a human brain can think of, thanks to this power of augmented intelligence and machine learning. Secondly, we know a lot about genetic predispositions, but a lot less about when and how those predispositions play out over time. The probability that a certain trait is actually manifested is quite often dependent on the environment in which that living thing finds itself in. Genes are the cards that an individual are dealt, but nature is the one playing that hand, and we're learning more and more about that interaction every day. And this is going to result in better, less expensive, and more informative tests for us and our pets. Changes in the way the physical structure of DNA, called epigenetic changes, ways of tracking which RNA are being transcribed in a cell under certain conditions, called transcriptomics, even ways of measuring the breakdown products of cells to assess their health and other important characteristics, which we call metabolomics. These are all just coming of age and becoming practical tools that we can use to help our dogs be the healthiest and best that they can be. So the future is exceptionally bright, and science is leading the way to knowing more and with greater confidence about the diseases and conditions of our beloved dogs. I hope you can feel the enthusiasm that I live with every day about the world that we inhabit and the brightness of our future and the future of our dogs. And this is a picture of my heart dog, Bridger, who's actually at this moment laying underneath my desk. He is a therapy dog. He is participant number 1199 in the Golden Retriever Lifetime Study, and he's the best friend a guy could ask for. He is almost 12 years old, and I hope that he'll be a part of my life for many years to come, and I believe he can, thanks to the kinds of tests you'll be hearing about later through the day. So thank you again for choosing to spend your time with us during the Canine Health Summit, and of course, I'd be happy to answer any questions you might have. Thank you. And Lisa, I think I'm ready for questions.
Thank you, David. That was a very interesting presentation. I learned so much. 6 feet of DNA strand in a singular cell. That's amazing. And the video was beyond cool. And also why that human-animal bond is so precious to us and why we love our dogs so much. So thank you for all of that. If you want to ask a question, just go to the Ask a Question box and put your question in there, and we will get it to David. There is one question so far about, it's about Embark. So where is Embark going in the pet care and wellness space in the future?
Well, I don't know how many people know the history of Embark, but when Adam and Ryan Boyko started it in 2015, it was really one of the originators of this intense science around genomics. They were just catching the original wave. And as a result, we have the largest database of DNA of dogs as we know it in the world, with over 2 million samples in our biobank. So that repository is an incredibly valuable a thing that we get to use, an asset that's at our benefit, but also intentionally for the benefit of the dogs that we serve. So where are we building off of that foundation? And I think I kind of foreshadowed it in the last slide. We are rapidly investigating and hoping to launch multiple tests on all these ways to better understand your pet, because we think at the heart of the human-animal bond is this need to nurture our companions tightly. And that means to better nurture, we need to better understand. And the more we can understand our pets, the better we can care for them. So foreshadowing our future directions, we hope to help pets, help breeders, help owners better understand their animals that they're working with, because it doesn't really matter what we come up with. As long as you better understand, you can manage the treatment of those animals better. So that's sort of where we're going.
Great. Thank you so much, David. We are now up to our first 5-minute break between sessions. So we will see you all in the next session at 11 a.m. with Dr. Jenna Dockweiler, and it's Canine Genetics 101 and also puppy DNA health testing. So see everybody in a few minutes. Bye.
Thank you.
Thank you.